Welcome everyone to our IXPUG webinar. Today's topic is Impeccable, a dream pipeline for high throughput virtual screening, or a pipe dream, an intriguing title. Our distinguished presenter is Shantanu Jha, Chair of the Computation and Data-Driven Discovery Department at Brookhaven National Lab and Professor of Computer Engineering at Rutgers University. His research interests are at the intersection of high-performance distributed computing and computational and data science. Shantanu leads the RADICAL-Cybertools project, which are a suite of middleware building blocks used to support large-scale science and engineering applications. Among his many accomplishments, Shantanu is the recipient of the 2020 Gordon Bell Award. Shantanu, please begin.
Thank you, Sherry, for that very kind introduction, and good morning, everybody. It's my pleasure and distinct honor to speak to you today about Impeccable, which is trying to bring the advances in scalable computing with machine learning and AI to hopefully revolutionize the way we design drugs. The context is as follows. I hope everybody appreciates the fact that drug discovery and design is a very complex and expensive process. Different drugs have different costs to develop, but on average, the industry standard, industry accepted numbers are it takes approximately 10 years and billions of dollars to develop a single drug. The process is such that it's simply not possible to do an exhaustive search over all possible drug candidates for a given target. The number here, 10 to the power 68, is a number that gives us all pause to think about the space of possibilities. Now, when you have such a large space of possible drug candidates, as mentioned, exhaustive search is simply not an option. So you have a process which is shown in the schematic on the right-hand side of a set of stages, a pipeline of drug discovery. Namely, you start at the top with a very large number of possible compounds. You take them through a computational method, which analyzes them, but only promotes the most promising candidates. So as you can see, you go from a very, very large number to a reduced number, and you rinse and repeat, you iterate this many times until you get to, let's say, the last stage downstream, where now you've got the most promising candidates, and therefore you're able to, on the select few set of candidates, able to employ very computationally expensive methods, but ones that are very accurate, ones that are likely to give you, let's say, very reliable results, and you only put forth the most likely candidates. So this is traditional high throughput virtual screening, and this has been around for some time, a decade plus, with a lot of innovation ongoing at the individual stages and coupling the stages together so that the whole is greater than the simple sum of the parts. What is different now is, can we take these methods and continue to improve them, but improve them in ways that bring machine learning and AI into the fold? So we continue the simulation or the other computational methods, but we enhance each of these methods with an AI-based approach. And I'll elaborate on this very soon. If we are able to do that, are we then able to select better drugs, or select drugs faster, or just go through the space much, much more quicker? So the question really is, if we're able to enhance our methods with these AI-driven approaches, can we do bigger, better, faster drug design? And that's the question motivating our work. The challenges in order to answer this as hopefully a definitive yes, or maybe a possible no, is to then think of what are the computational challenges, what are the performance issues in order to do so. And just because this is a performance user group. This is about computing. With this motivation, I want to kind of tell you the following primary upshot stories of this talk. The first is that when you're able to couple these traditional computational methods with machine learning, you are able to do bigger, better, and faster. Details coming. But importantly, this imposes a new set of requirements on supercomputers that we've historically designed and used. And it is the systems software challenge that really needs to be rethought. The system software needs to be rethought so as to support what we call heterogeneous and scalable workflows. And what IMPECCABLE is the name of a project, I'll speak to that in a second, that shows the benefits from advances in the methods with the enabling software, system software, as well as the scalable workflows. So that's in a nutshell what this talk is about, to give you our experience of how we're able to radically change this drug design pipeline. Now, the context for this, as many of us, all of us know, unfortunately, is COVID. And specifically, this work has been done in the context of DOE's National Virtual Biotechnology Laboratory, which brought many facets of the DOE together for many different parts of the national response to COVID. Everything from epidemiological modeling to supply chain understanding. But what we were involved in was the third bullet, namely the supercomputing and artificial intelligence for design of targeted therapeutics. But this is the DOE that we're talking about, which specializes in designing high-performance scalable solutions and methods, software, hardware. And the challenge was, could we put this expertise, decades of expertise, leading expertise, to the service of the nation and the community as a whole to try to advance therapeutics for COVID. And this all came together very quickly, as you can imagine, literally started running days after the WHO declared COVID as a pandemic. And again, a long story short, we were up and running within weeks and generating results and data in ways that I will show you. Just a kind of retrospective, we're 18 months since, approximately 18 months since the start, and whereas we made a lot of ongoing efforts and we made lots of advances, including proposing antiviral drug candidates, this is very much ongoing work and by no means has the final word been written. However, we're still very lucky and honored to have been given this distinct award from the US Secretary of Energy in 2021. Okay, so let's take a step back and look at the drug design problem as a specific incarnation of the computer systems challenges and perspective. And I don't think I need to tell this audience that the trends that have driven performance and progress in computing for the past many decades, namely Moore's Law and Dennard's, have, for all intents and purposes, either come to an end or have, as more accurate, has modified, has changed the way we can extract performance. And there are different ways of looking at this, but we all know we're in this flat curve region, flat plateau region, where just doing nothing will mean that our codes will actually run slower with subsequent generations and subsequent architectural advances. Not to mention the many challenges that this imposes, that we have to modify our software every time there's an architectural change, and that's highly costly. The porting algorithms, porting are very complex scientific processes to every architectural change, non-trivial. So the question that arises from a systems perspective is, yes, we have to stop thinking about the performance of just a single task, a single simulation on this, if you will, changing landscape, changing architectural landscape. But can we now also think about deriving performance that isn't sensitive to the underlying architectural changes? Now, There are many answers to that, if you will, question, but one approach, which I'm sure, again, many people will be familiar with, is to take the benefits of traditional high-performance algorithms and methods, over which billions and billions of dollars over the past decades have been spent and have been used to advance the state of the art, and enhance them with AI-driven methods. This is important. I'm not proposing we say substitute HPC and just use AI, but trying to say, can we supplement HPC algorithms and methods with the many amazing advances, rapid advances in AI-driven capabilities? And the question that really arises is, can AI-driven approaches enhance the effective performance of traditional simulations? By effective performance, I mean the scientific insight that you can get for, let's say, a certain amount of computational capacity, computational budget. And so that's the kind of the question that arises. And in order for this to happen, we have to step back again and appreciate the different ways in which AI will enhance, will supplement HPC simulation. So we call this concept learning everywhere, where you're coupling machine learned methods to traditional HPC methods. And whereas there's a very large taxonomy of the various ways this can happen, let me distill this down into three primary ways in which machine learning couples to HPC. I'll call this ML X HPC, where X is in, out, and about. Let's start with the simplest, ML in HPC, where you take an HPC simulation and you use that to train a machine learning model. And in some ways, you use the simulation to train the model, but the model itself substitutes part of the simulation, or in some cases, the entire simulation, as an extreme case of the in. So in to encompass the entire HPC. So let's talk a bit about substituting part of the configuration of the simulation. And the best example is, if you will, to train use machine learned models to understand, as is often the case for the potentials to understand compute the Hamiltonians of in particle simulations, particle dynamics. So that's a very simple but a very powerful example of ML in HPC. The second category is ML about HPC, where you run many simulations, but as the data from these simulations is being generated, you are training a model, not substituting the HPC, but you're training a model to steer, to guide these simulations, ensembles of simulations. So these become smart ensembles that are then, if you will, guided through phase space to be able to, let's say, do more intelligent simulations in the right parts of phase space. And this is what we call ML about HPC. So you've got the ML working around the HPC simulations, but guiding them. And then there's the last category, which I'll be very quick and talk about, ML out HPC, where the entire campaign is actually being monitored by a learned simulation, reinforcement learning, active learning, to try to determine the optimal set of experiments, computational in this case, to perform, right? So lots of details in each of these categories, and I invite you to look at this, what we call the Learning Everywhere trilogy set of papers written by myself and my colleague Jeffrey Fox, where all of these are elaborated with significant examples. Okay, so here we're going to talk a bit about ML in HPC and ML about HPC. So to give you some very simple examples of ML in HPC, there's the global circulation models central to many climate studies, including the recent IPCC. And just to give you a kind of a sense of what the power of these learned surrogate models that substitute part or entire simulations, on the extreme left-hand side, you have what are called the global circulation model real simulations. In the central column, you have an emulator trained using data from the simulations. And in the extreme right-hand side, you see that the emulators, the surrogates, have very strong, very high predictive capabilities. They are almost able to reproduce results of the simulation without loss of generality, or in this case, performance. However, The important point is that the cost of being able to reproduce these simulations once trained, important to point out, is significantly low. And just look at the bar on the extreme right, left-hand side, beg your pardon, and you'll see that the global circulation model is 1 billion times faster than the real simulation. I repeat, 1 billion times faster than the real simulation. And this is not an isolated or a singular event. This capability of using a surrogate for substituting parts of a simulation has now been repeated in many different domains. And this is another example, in this case, coming from quantum molecular dynamics. And lots of details here, but as you know, in quantum molecular dynamics or molecular dynamics in general, the bulk of the computational cost is to compute the forces acting on the atoms or molecules so as to be able to compute the next position and velocity at the next time step. Now, this can be computationally intensive so as to occupy up to 80–90% of the total computational cost in these systems. So the question is, can you actually use a learned model to substitute the calculation of either the quantum Hamiltonian or whichever Hamiltonian you have? And this paper shows that you can actually use the simulations to train a model that then actually computes the forces on the particles. And when you're able to do so, lots of efforts in training that model, you can then run the simulation with a learned model computing the forces, positions, and the velocities, as opposed to a brute force calculation of the Hamiltonian. And you can start doing wonders. There's, again, 1 nanosecond for 100 million atoms is kind of a gold standard, and you're able to get that once you use this learned model. Again, details in this paper, a highly impactful paper by people like Roberto Carr and others, and trying to show you the way in which machine learned capabilities, when embedded inside traditional HPC, can really improve the effective performance of HPC. Okay, so up to now, you've seen the importance of creating methods and approaches that are not susceptible to the architectural churn. You've seen the promising approach of AI-driven HPC algorithms to enhance the effective performance. And I hope I've given you a bit of a flavor of how some examples of ML inside HPC work. But the important, I think, generalization is that once you have machine learned capabilities along with HPC capabilities working collectively together, you no longer have a single task, you no longer have a single uniform homogeneous simulation. You've got a very complex mix of many different distinct tasks running. And this is what we call a workflow. And in particular, because of the distinct nature of the many tasks, you've got heterogeneity. And because they're kind of changing dynamically, they're often adaptive in nature. So how do we think of workflows that are heterogeneous and adaptive in the context of drug design? Enter IMPECCABLE, where IMPECCABLE is a multi-stage campaign with many distinct workflows at each of these stages to try to improve the overall throughput as well as quality of the drug discovery campaign. It's important to go back to that schematic early on and say that the outline is very similar. You've got multiple stages, but each of these stages is now machine learning enhancing traditional simulations. So we've got, in this particular case of IMPECCABLE, we have a high throughput ensemble docking, a very common method, a common stage in the drug discovery pipeline. We have that here, we call that WF1, workflow 1. But now we're going to enhance the docking capabilities with a surrogate. We have downstream at the very other end, binding free energy calculations. These are the industry gold standards upon which drug candidates are selected or discarded. So we have binding free energy calculations, which we once again enhance using machine learned approaches. So I hope you get the flavor that what we're trying to do in Impeccable is take traditional methods, obviously implement them at unprecedented scales, which I'll talk about. But as we implement them at unprecedented scales, we also enhance them with these AI learned approaches. So we get traditional performance gains, but we also get effective performance gains. So to give you a couple of details of the individual workflows involved, let's go to the first part, the ensemble docking, where you've got many different drug libraries, in this case, a library of 6.25 million candidates that have been curated by either a company or by academics, people who have good reasons to believe that this is a nice set of starting points, 6 million ligands approximately, to try to find a drug that will bind to a specific target, in this case, a target which could be a receptor on the S virus of the coronavirus, the spike protein of the coronavirus. Let's think of this computationally. So now you've got 6 million ligands in the library, but you have 32 different targets on the spike protein. Now what happens is that this is a computational process that is very sensitive to the specifics of the target and receptor. And what the top left-hand figure shows you is that the time that it takes to run through these 6 million ligands for each of these 32 targets, and without getting into details, you see there's a very broad distribution of time taken. The library, the time it takes to go through the library is very sensitive to the specifics of the target. Furthermore, if you take any one target, any one receptor, and you look at the time it takes for the 6 million drug candidates to be docked against each of these, you find that the time within a given target receptor for the 6 million drug candidates is very broadly distributed, hence the long tail for each of them. So this is telling you that these computations are truly heterogeneous in the temporal sense, and you now have, as you can imagine, an immense load balancing problem. So remember, I talked about heterogeneous and adaptive workflow. Here's an example of the heterogeneity of the workflows in a temporal sense. Now let me give you a second, and this is just showing you how the load balance has to be managed. In this case, over a couple of hours, you can see that when you're able to do a load balancing, you're able to kind of get the docking rates to fluctuate around a factor of 2, number of hits, number of dock hits at a given instant of time, whereas in the previous slide you saw factors of 10 and 100 of variation. So without getting into details, you start seeing some of the challenges of these underlying workflows, the system software to support these the load balancing, and I'll come back to that in a short while. Remember, I said each of these stages is operating at scale. Gave you some examples on the previous slide, but some of the real requirements are to run these methods at scale so as to train a machine learned model. And what the schematic on the left-hand side shows you is how you would do docking in a historical, traditional sense, where you take a library, in this case of 6 million drug candidates, you dock them, you take the top end scores, and let's say top 1%, and then you would go off to either do further in silico experiments or real laboratory experiments. However, the workflow now is as given in the column B, where you take a large amount of the libraries, you do enough of the docking so as to create a machine learned surrogate, which then aids you in determining the likely scores of the rest of the candidates. And there are lots of details presented in this paper. The point that I would like to convey here is that when you're able to introduce surrogates into your docking workflow, you can, without loss of accuracy, be able to span or sample a factor of 10 more drug candidates for a given computational budget. Factor of 10, effective performance. Now, this requires a lot of care and caution to make sure that your accuracy of prediction is within, let's say, the acceptable error. And in this case, the acceptable error is 1%. So you don't have false positives or false negatives, lots of details. But the upshot is you can use your scalable approaches to train a model that, once trained, allows you to do sampling of your docking candidates, drug candidates, beg your pardon, 10 times more efficiently for a given budget. So remember the 10 to the power 68 space we have to cover. Here's a modest improvement in the rate at which you can sample through or span through the possible space of candidates. That's one example, but another interesting example is when you can start, before you compute the binding free energy, if you can pre-process these stages with a machine learned model that steers the many simulations so as to be able to sample the space of energetic and conformations of the bound state of the drugs that you just selected through the docking phase. And so as those drug candidates that you've selected, as they are bound to specific targets, can you get their conformations and sample those conformations correctly? And long story short, when you use a machine learned model to try to sample these configurations, what the bottom right-hand graph shows you is you're able to do this factor of 10 to the power 2, 100, to 1,000 times better than if you just did a vanilla traditional simulation. Okay, so even though there's lots of details to be still covered, I hope I've given you a flavor, if not a tease, of the fact that when you couple your traditional stages of the pipeline with machine learned models, you're able to, for each stage, get hundreds, 1,000 times improved effective performance. Okay. Now, it's not just about each stage, it's about coupling them all together. And that's where Impeccable comes in. So you've got this, if you will, 4-stage, 5-stage pipeline, but you want to run them as a whole. And before I get into the details of how we run each stage at scale, but run all of the stages together, just want to remind you of the advantages of an integrated approach, as shown in the figure on the left-hand side, where you can see the red lines represent binding free energy calculations without any machine learning processes aiding your calculation. And if you see this, you see red lines are high, red points are high, higher is worse, lower is better, whereas the black dots at the bottom show the binding free energies once you've got machine learning processes enhancing the binding free energy calculations. And so that shows you that you don't want just one calculation as a standalone stage, but you want to really couple them all together in order to get better output. So remember, I said bigger, better, and faster. And so I hope you're now starting to see the better aspects of it as well, in addition to the bigger and faster. Okay, so why is all of this a challenge? Why is this a challenge that I stand here in front of you talking about and trying to say this is truly moving the needle in terms of our capabilities of the community? Well, there are many reasons why doing all of this is challenging, in addition to the methodological challenges of coupling machine learning capabilities with HPC simulations. I hope I've convinced you that at each of these workflows, at each of these levels, are very distinct workflows. Sometimes you just have docking function calls, traditional Pythonic function calls. Sometimes you have very large MPI simulations, single node, multi-node, just traditional honest to goodness physics-based simulations. And in between, you have machine learned and simulations interacting and kind of growing together. So there's a heterogeneity of task types that you've got to manage. And it's not just heterogeneity in the task types. they are also very different in the cost, if you will, of processing a ligand. Between the first stage and the last stage of the impeccable pipeline, you've got a 10 to the power 7 orders of magnitude difference in the computational cost as measured by node hours per ligand. And you've got to manage this very broad range of costs in addition to the heterogeneity. So no surprises, our supercomputers were not designed for the scale, for this heterogeneity, and for this kind of diversity in workloads. And this got to all run together under the same single quote unquote job in order to get the benefits of something that I showed you on this plot on the left-hand side. So that's where the research challenge comes in. And just to give you another dimension of heterogeneity, there's the workload heterogeneity, there's the temporal variation heterogeneity, but then there's also the set of platforms in order to be able to achieve the kind of science we're trying to achieve. We also have to work on a very large set of supercomputers, everything from Summit, a DOE machine at Oak Ridge, to Frontera, an Intel-based machine at TACC, to machines in Europe, SuperMOOC at the LRZ in Munich. Okay, so if you now start thinking of where we are, the last part is to really think of extreme scale programming and system software to support the kinds of heterogeneous and adaptive workflows we've talked about, and ask the question, what are the implications for the system software to evolve in response to these methods that bring AI-driven HPC algorithms together? And without going into details, my own research revolves around RADICAL-Cybertools, the middleware building blocks to enable such workflows. But importantly, this is part of a very large, ambitious DOE ECP project, exascale computing project called ExoWorks, where we're trying to bring many different tools and technologies together to support precisely these kinds of workflows to be able to work on DOE's current and future machines, including the Intel-based machines that will hopefully hit the floor sometime in FY21 and 22. So please do look up ExoWorks and you'll get a sense of the cyber tools and other technologies that are coming together to support the kinds of workflows I've talked about. Now, I just want to give you one example of the kind of sophistication that these workflows require in order to run what I talked about, the heterogeneous mix of tasks. Sometimes they're function calls, sometimes they're non-MPI simulations, sometimes they're MPI-based simulations of a varying temporal dimension as well, sometimes a few seconds, milliseconds, sometimes many, many thousands of seconds. And the tool in particular is called RADICAL-Pilot with a master worker overlay. So what's the concept here? So just try to give you an example of the system software that we've developed to support Impeccable. Think of a pilot job as an empty job that goes on to through the queuing system, regular batch queuing system, and acquires all the nodes, could be the entire machine, could be a large fraction of the machine. And once it acquires the nodes, like any regular job, but without a workload assigned, it hands over control of these nodes to the application, the workflow, the campaign manager. And it is now the campaign manager, the application, that takes control over these nodes and can slice and dice them temporally and physically in any way that it so chooses or as is needed. And so in order to do this, in order to be able to place the right set of tasks on the right set of nodes at the right instant of time, what we do is we create these master tasks, which go out, create a communication and, if you will, scheduling overlay on sub-partitions of the resources. And once this overlay has been created, we then place worker tasks onto in conjunction with these set of masters, which then are able to, they are so designed to be able to place set of distinct tasks, in this case, MPI and non-MPI tasks, appropriately. But the world is more than just MPI and non-MPI, if you will, simulations. There are also a set of, if you will, function tasks, which need to really, in the sense of the Pythonic function calls, that need to also be managed. So you see for the first time, within the same job, this complex interplay and management of MPI, non-MPI simulations, and the Python function calls, as well as the machine learning function calls, all managed by the system software capability of RADICAL-Cybertools. And once all of this happens, you're able to manage the workflows I gave you examples of. So now let's step back and just understand and appreciate the scale at which this capability has to be managed, has to be executed to try to support the kinds of workloads and workflows that IMPECCABLE needs to address to solve the drug design problem. So lots of details, but in red, I hope to focus your attention on Frontera, the machine at TACC. And what I want you to focus on is the second and the third rows, where when you can use somewhere around 8,000 nodes, which is almost the entire machine, you're able to do docking at approximately 150 million docks an hour. Now, this is a factor of 2 or 3 more than any other published result that we know of ever. And that's because of the system software capabilities that allow you to manage these very broad range of function calls. And this obviously requires managing 400,000 concurrent tasks. So you start seeing the importance of system software to support the kinds of workflows we're talking about. And this is just to show you that we get this kind of concurrency, 400,000 tasks per second, at any given instant of time, without any loss of resource utilization, 95+% almost indistinguishable from 100% utilization. So that's just giving you an example of how every core, every node of a machine like Frontera has been used for the IMPECCABLE workload. But that's not enough. You have these machine learning driving the simulations, and this particular workflow was done on Summit. And again, without getting into all the details, what I want to highlight in the red bolded box is to show you that these workflows can use every core of every node and every GPU of Summit. So that's about 26 to 27,000 GPUs and approximately 240,000 plus CPUs. And each one of these can be used to drive the machine learning driven HPC simulations. Okay, and the important point is that in order to be able to run this as a single campaign, what you end up having is the need to have variations on a theme. You've got the same pilot job model, but you now need customizations that are hidden away, abstracted out from the application, but are then customized for the specific machines, whether they be Summit, or whether they be Frontera, or they be some other machine. And that's what the power of the RADICAL-Cybertools abstractions is it gives you this concept of a pilot job that's customized and specific to each machine, but without exchanging or changing the interface and the execution model for different applications on different machines. So trying to bring everything to an end, I hope I've conveyed to you some of the system software challenges we've had to overcome for this very interesting set of applications, these novel methods at unprecedented scale. So what has all of this led to? So yes, we've computationally docked more drug candidates than anybody else as far as we know, 100 billion docking calculations. We've done hundreds of thousands of traditional molecular simulations, many of these binding free energy calculations enhanced by machine learning. We've consumed, just to put things into perspective, 2.5 million node hours using Summit as a baseline. That's about 30 days of all of Summit, obviously over different machines, peak performance of 8,000, 4,000 nodes. And what this has led to is at least one, possibly more drug candidates, antiviral drug candidates that have been identified and are now in experimental synthesis phase and are being investigated in the lab for possible clinical precursors to clinical trial. So obviously we've made an impact on the original mission, namely impacting SARS-CoV-2 therapeutics, but obviously this impeccable pipeline is representative of any design problem. And then the question is, what beyond COVID can we use for? And obviously, as you start operating at scale, you need many more methodological innovations. And I'll just speak to one, namely, how do you determine the optimal use of resources for each stage? So remember, right now we hand tune arbitrarily, say this one doesn't go forward, this drug candidate does go forward, but we need to build up the mathematical formulation so that the multi-stage pipeline is guaranteed to give us an optimal use of constrained, limited computational resources. So that's just one of the methodological advances that Impeccable is trying to embody. And I hope it also gives you a flavor of how Impeccable brings together domain sciences, mathematical sciences, and computer systems professionals. So in summary, drug discovery is a complex and expensive process which requires methodological, mathematical, and infrastructural progress. And Impeccable represents a first but very important approach to using scalable computing and machine learning to enhance the effective performance of the drug discovery pipeline. It is impactful. However, it is still the first generation, and Impeccable has to evolve as these methods evolve, as these methods become more powerful, more general. They have to evolve. It has to evolve to support these different methods at scale. And I've hopefully given you some perspective into the system software challenges and ecosystem that must evolve in order to support these kinds of problems at scale. And the point will be, as the methods evolve, the system software challenges will continue to evolve. And what we're bound to see is an important set of advances in these adaptive runtime systems that can run on clouds, on extreme scale supercomputers to support the very broad range of heterogeneity and adaptive workflows that I mentioned. With that, I just want to mention this is work done by a very large number of people at a large number of DOE national labs, universities, as well as international partners. And thank you very much for the opportunity to present. I'd be glad to take questions.
Okay, thank you, Dr. Jar. This was really amazing work, and I'm very happy to see this talk at IXPUG. So just a reminder to our attendees, if you want to ask a question, you can raise your hand and we can unmute you, or you can type your questions in the question box and we can read them out, and Dr. Jar can answer the questions. Okay, while we're waiting for the audience to possibly come with the questions, I do have a question for you. So what is in your experience when you were running, for example, at 7,000 nodes or almost 8,000 node scale on Frontera, what was the biggest performance challenge for you?
Yeah, thank you. This could be a lecture unto itself, and for a while I thought I'd just talk very deep about Frontera. We were in the midst of writing a paper, so happy to share that as soon as it's ready. But the single biggest performance was related to the fact that when you have 400,000 independent Pythonic processes, that's not going to be very kind to the file system, as you can imagine. And so we had to work extensively with colleagues at Frontera, at TACC, to try to manage that performance degradation that happens when you have 400,000 plus independent Pythonic calls. So that was one, but and I would say, great tribute to our colleagues at TACC to have helped us manage that. But once we got that, As you can imagine, and as I showed in one of the slides, the time distribution of these individual function calls was very, very broad, from seconds, sub-seconds really, to many, many minutes. Once you have that very broad range of a very dynamic set of tasks, getting the load balancing so as to still get that 95+% that I showed you of resource utilization, that for us was a scheduling and a performance challenge that we had to overcome with our tools, with our system software. And last but not least, managing the failures, because some of these are, as you can think, these are physics-based approaches. So sometimes things blow up, so function calls fail. So managing failures as they often happen in scientific calculations, not for systems reasons, but for scientific reasons. And so to not bring the whole set of calculations down, or more likely, not to have nodes that on which there was a failure be, let's say, out of commission for any longer than the absolute necessary. So in summary, file system, load balancing, and making sure that we were resilient to failures. These were the top 3 challenges.
Right, okay, thank you so much. So I think we have a question from the audience. What kind of MD simulations you're using to calculate binding free energy? For example, steered MD or something else. Also, how do you parameterize each drug molecule? Do you have a pipeline for parameterization?
Great question. So the answer to the first part is, in terms of MD engines, we use both AMBER and GROMACS and NAMD. But for the ones that I talked about here, I've obviously glossed over some details, but even within the binding free energy, we have 3 different types of binding free energies. So one of them uses AMBER, one of them uses NAMD. For what you see clearly, the question is being asked by somebody who understands molecular dynamics very well. It's thermodynamic integration. So it is a steered MD, alchemical perturbations. But that's just one of the 3 different types of binding free energies we use. In terms of parameterization, yes, I think that was done offline, not part of this workflow, this campaign. We obviously used well-defined parameterized both MD as well as docking calculations. And I should mention that all of this is published, and I am remiss in not placing the links to the papers at the very end for simplicity, but I'll share the slides, and the references are in the slides. So you'll be able to find more specific details for your taste.
Okay. I think I'm getting some of the questions.
Yeah, I have a question. So we have worked in a very large team, and was it really challenging for you to share the data across the organizations and across the cluster? How did you manage that?
Yes, sharing the output of these, if you will, simulations, these workflows, these campaigns was highly non-trivial. So there's 2 components to that. One aspect of the sharing was we took data that has been generated, and not just by us, but by other members of the community. And there's an NSF effort project called MOLSI, Molecular Science Software Institute. So MOLSI has been, if you will, a custodian of community-generated simulation data. And I urge you to go to molssi.org, and you'll see that they've got a COVID-19 repository where they've allowed and enabled the community to share simulation data. And I'm a co-PI of that project, so would be glad to address some of those specifics there. But that was one modus in which a lot of activity data, not just ours, but the community globally shared. Specifically, aspects of the docking phase and the machine learning data were shared by Argonne National Lab through a capability called Petrel that is supported at Argonne National Lab. So these were the 2 primary modes of data sharing. Now that's just kind of trivial data sharing, but as you know, you want to share data, you want to share metadata, you want to share more insight, and obviously we are limited in the upper levels of sharing that we have. And this is where, once again, I'm hoping the community can provide solutions so that we can take some of the capable data that we've generated through really highly expensive simulation. 2.5 million node hours by any account is worth $10 million plus of computing. However you measure it, it's, in my opinion, worth much more. But that's a non-trivial amount of computational cost. And it would be a shame if, as they say, the data went to die on our file systems. The data should really be used. But that's a very different set of concerns, expertise, and effort than my team has. So would be very glad if somebody would to step up and to talk about how we could expose that to the broader community. Sorry.
Do you intend to provide this data to the general public?
Yes, absolutely. This is all open source and funded by taxpayer science projects, and almost by definition, open data as well.
Thank you.
Sure.
I guess I'll ask one more question because I'm curious. So when you're running these, and I saw a bunch of machines, right? DOE machines that you're running on. How do you find the library support or even performance analysis tools?
Yeah. So great question, and I think it's important to appreciate the many days, weeks, and months of priming and preparation and processing that it takes before we can run at scale. I think you'll appreciate not only that we don't go to 8,000 nodes or 4,000 nodes on the first attempt. The rule of thumb that I give my students is factors of 2. So start at 128 and then go factors of 2. So clearly the 4,000, 8,000 nodes represent the high water mark, but by no means our first foray into wetting our feet. Now, the journey from, let's say, 128 nodes to 8,000 nodes is a fraught journey. And sometimes the tools and the support for performance analysis is adequate because we're trying not to improve the performance of individual executables, because the AMBER community has done that, the NAMD community has done that, and obviously the PyTorch and the TensorFlow community has done that. What we, as in my team, are really concerned about is the notion of collective performance. When you put all of these distinct tasks together, how do you manage to keep the performance what you'd expect it to be? Namely, if you run 10,000 or 1,000 separate heterogeneous tasks, each task is doing what it should be doing without stalling on another task or stalling on the, let's say, system performance. So in simple terms, we don't try to tune the performance of individual tasks because these are often optimized for the supercomputers in discussion. We are working to try to make sure that resource utilization, collective performance is on hand. And with that long-winded background, I come to you and say it's a true labor of love and feat of the graduate students and the research scientists in the team that we're able to get that performance, because we have very little, if any, form of tooling support to think about the collective performance of the workflows. And so this would be something that I would be forever indebted if members of the community, members on the call, would find this to be a research opportunity that we could all collaborate with between the industry, between universities, and my team at the national labs. I hope I answered your question.
Yeah, you did, and I think that's exactly what I wanted to know, because I think at Intel we do a good job of VTune and others to help you optimize that performance of the binaries at executables. But I think I think we'd be very happy to have a conversation about how to do ensembles and do pipeline performance analysis. So maybe we'll have you on for a call internally and learn from your experience. Yeah,
I really appreciate the opportunity to discuss further, and I think people like you and your team would be far, far better placed, given the experience you talked about, VTunes and others, to raise that to a higher level, move away from a single executable, but collective performance of an ensemble or a set of heterogeneous tasks. So yes, I look forward very much to that.
Sure. Any other questions from the audience? We have a couple of minutes for the hour. Okay, I guess that's it. Thank you so much, Dr. Shah. This was really interesting, and I really enjoyed it. So we'll, for the audience, we'll share the slides and put up a recording of today's talk on the IXPUG YouTube channel, as well as on the IXPUG website. And yeah, please share the link and watch it later. And I think this is great, and this is where I think HPCs, a lot of HPCs heading, you know. So this will be it's a really good talk. Okay, thank you so much, Dr. Jha. Thank you to the audience for attending, and we'll see you for the next webinar.
Thank you very much for the opportunity. Look forward to further discussions. Bye-bye.